Hello, I'm Kevin Davis, editor of BioIT World, back here at the Rhode Island Convention Center, where behind us CHI's Exploring Next Generation Sequencing meeting is going on. But dragged out from the exhibition hall is Mike Cariaso, who is a senior scientific consultant or something like that with the BioTeam. Mike, welcome.
Thank you very much.
Tell us first a little bit about the BioTeam, which is having a kind of a bit of a field day here, I suspect, with this hundreds of people desperate to know how to manage all of the reams of data coming off their sequencing machines.
We're definitely in our element. It's kind of fun to be here and just watch it all happening, talk to people who are actually working with the data, developing the machines, the full spectrum. But this is our native environment. It's wonderful.
So the BioTeam is a group of a number of pretty smart BioIT type professionals. What do they do, and what are your key areas of expertise?
Life sciences consulting, I think, is the broadest umbrella that covers it pretty well. A lot of expertise in next generation sequencing, understanding this is really an area that continues to expand, and I think it's a little bit underserved. We have a lot of experience as well with large-scale storage systems. For about a decade now, we've been building clusters, data pools, all the rest, as a way of keeping track of data. Because very often we're talking about massive data sets, large computes, and these are things that are happening in somewhat small wet labs increasingly as all this next-gen sequencing trickles out of the large sequencing centers and instead comes into university environments instead.
So you and your colleagues are working with both with pharma companies, biotech and academia, really across the board?
Gov, Mill, EDU, you name it. We've had a wonderful spread. In fact, as one of the more recent people to join the BioTeam, I've really been impressed by how many other environments I'm getting into because of all the work we've done in the past.
And what did you do before you joined BioTeam?
Many things. I think Lawrence Livermore, I can start with.
That you can talk about on camera.
Okay, but maybe the one that's most interesting, I think, here is I tested myself, in fact. The way that there's now 23andMe and lots of this sort of personal genomics industry sprouting up, and it's very much related to the work that's going on here. I was sort of an early person poking around in the corners of all this and trying to learn more about myself. So I had a blood draw and sent off some blood to a lab and got myself running a microarray. And this is now what's available for, in the case of 23andMe, a $400 service. But it got me an early taste of all of this and really sort of excited me about where genomics and medicine and health and technology is all heading.
Let's come back to that in just a couple of minutes. Before we do, let's talk about one of the projects that you're particularly responsible for, I guess the chief architect of WikiLimbs. What is WikiLimbs and who's using it, and how is it helping people working with Illumina and 454 and other next generation sequencing platforms?
Sure. The next-gen sequencers are really hardware devices, and they're here producing just oceans of data. And you need to somehow keep track of what did we run yesterday, what sample went into that run, who processed it, all the rest. We need some sort of what's called a LIMS, Laboratory Information Management System. It's a database. But we're trying to build a database for an industry that's just changing literally every day, and every customer has very distinct needs and ways that they're using their data. And so everybody's familiar with Wikipedia at this point. Wikipedia is written on some software called MediaWiki, and we're using that and some third-party components, in particular Semantic MediaWiki, to build a very scalable, very flexible database that's being used for both storing the data and then initiating some of the downstream analysis, visualizing your results, making sense of all of it.
Who are some of your early adopters?
Let's see, a Navy group in Rockville was the absolute first one. In fact, it was built because they had two 454 machines sitting side by side and no real way to keep track of which machine had done which run, where were the results now. That's gone off to National Cancer Institutes, Emory University. We're actually showing off quite a bit of the system in use at Cold Spring Harbor National Lab. Cold Spring Harbor.
Laboratory,
yeah. And I'm probably forgetting plenty more, but there's quite a few out there in the field. We get excellent feedback. In fact, James Van E from Cornell's Sequencing Center I just ran into, and it was a nice chance to reconnect with a very happy customer, it turns out.
So the kind of customers you're looking for would be sort of core labs perhaps, or medium-sized centers. I would imagine the big genome centers probably have their own infrastructure that they may not need this.
It's true that the Broad and all the rest just have tremendous resources available and very, very unique needs. We're trying to figure out best practices from the groups that we interact with, which tend to be maybe a step down in scale from that, and are in some cases core facilities, but not the JGI style sequencing scale. I think we got early exposure in that environment because the next-gen machines turned up there first, but now they really are trickling into smaller environments and we're able to leverage and reuse pieces we've built previously.
And WikiLims is sort of agnostic of platform. It doesn't matter which kind of sequencer. This is to really marshal and organize and plan the experiments.
So much so that it's not even next-gen specific in any sense. We're using it much for 2D protein gels, microarrays, all the rest. WikiLims approaches it and assumes that everybody's going to be different, not just between vendors, but between different releases. We assume that you have a file or a directory of files being produced, and that's what we're going to go in there. You're going to give us some way of pointing to where your data is. We're going to visualize it, analyze it, and make sense of what this particular platform's producing.
Let's just spend a few minutes in conclusion talking about one of your other projects, that's kind of a side project, but predates BioTeam, I guess, and that is SNPedia, which you are also the architect of. Tell us how you got into SNPedia and what the current status of that project is.
Sure. I had worked with, I'd actually worked at GeneLogic for quite a while and gotten some initial exposure with the Affymetrix microarray platform and understood enough of what was going on here, but it was all somewhat abstract. You're dealing with just sample X123. And I knew what the kind of data we were seeing and where this sort of felt like it was all going to head, and I wanted to make it a little more personal. And this is why it's now somewhat called personal genomics, but I wanted to know what my own DNA said about me. So along with a friend, Greg Glennon, we just started keeping track of all the SNPs we could find literature for and understanding what genotype has what consequence. And we needed a database to keep track of these SNPs we built. And that's the birth of SNPedia. So SNPedia is in fact a wiki database, and it's in some sense the inspiration for a lot of the WikiLims, but it's really been growing tremendously. We're having lots of traffic, lots of the next-gen sequencing companies and personal genomics companies are regular visitors. And increasingly it's being used not just as a database in and of itself, but for its real original purpose. It was always designed so that as a programmer, I could use it to compute and write tools and build tools. And now it's the back end for a tool called Prometheus, also available from the SNPedia site, that lets you make a very comprehensive report about what your DNA says about you, regardless of whether your data came from 23andMe, Navigenics, DecodeMe, or any of these other platforms.
So back to SNPedia. SNPedia has something like, you were just telling me before we filmed this, 4,000 SNPs out of 12 million or however many human SNPs there are. These are SNPs that have some sort of functional or putative functional impact on a trait or a condition or a disease.
Some connection between a genotype and a phenotype in the industry parlance. But yeah, we need to have, we could put all 12,000 human SNPs in there tomorrow, but if all we could say is, there's a SNP, it's on this chromosome, we wouldn't be doing anything that DBSNP or other databases weren't already doing. We're very much focused on the SNPs that tell you something interesting. And as a result, we have to track things from the very beginning. There's a certain, I think, tension in that the companies like 23andMe want to only provide information that's well vetted and confirmed in lots of journals. And the same is true for me. I don't really want to start responding or making behavioral changes or dietary changes until I have some confidence. And so I look for a SNP to have maybe 3 papers or so and really read over the papers and judge the science for myself. But SNPedia has to capture the first and the second one if we're ever going to know when the third occurrence of a journal citation comes up. And so increasingly we're just building up this database of genotype to phenotype correlations. Some are single SNPs, some are in fact multi-SNPs, some are based on indels and other variations.
But if I'm a scientist and I've identified a SNP with a phenotypic correlation, I can enter that information. You're not the only person who puts that in, right?
We would love it, and in fact we appreciate the folks who've done that so far. It really is. It's a Wikipedia-style community site trying to bring this information in, and we really have to rely on lots of experts in different disciplines adding to their part of the story. One paper comes out and gives us a certain expected result, but it's only done in the Caucasian population. I have to rely on somebody else someday coming along and confirming it in other populations or expanding the research.
And where Prometheus comes in is that if you've done one of these personal genomics tests from Decode or Navigenics or Seqrite or 23andMe, they're telling you information on a few dozen conditions right now, but the chips that they use, whether it be Illumina or Affymetrix, provides information on thousands and thousands of SNPs, many more of which are catalogued in SNPedia. So you can take your basic data available from all of those companies, and then Prometheus allows you to kind of get a much deeper drill down into the long tail of SNPs that you have that 23andMe and et al. Don't feel it's quite ready for prime time yet.
Yeah, it's certainly a peek into things that are not nearly as well confirmed yet, and that's just the necessary part of the science itself is emerging, and SNPedia is there trying to keep an eye on all of this. But regardless of who did the testing, most of them or all of them do release a raw data file to you. You're going to get back between 500,000 and maybe just shy of 2 million SNPs on some of those platforms. And we're able with Prometheus to read the raw file formats from all of these vendors. If somebody out there has a format we're not yet supporting, we just need to know about it. I'd be very happy to add it. But it's a way of really drilling into all of the information we understand. And I think it does an important thing in that it separates out what do we know for single SNPs, and how that one SNP might directly lead to a certain disease, for instance, ApoE4 as a SNP related to Alzheimer's, but you may have 25 other SNPs that all also relate to Alzheimer's. And Prometheus, because of this very broad view of all of the different pieces we know, gives you a way of putting more of your information into context and understanding, I think, the bigger picture of how a small change in your genome may or may not have an effect on a disease.
Well, Mike, thanks for taking a few minutes from your busy duties at the Expo booth, and good luck with BioTeam and with SNPedia and all of your other exciting ventures.
Thank you very much.
I'm Kevin Davis, BioIT World, signing off. See you next time.